Hello and welcome to this special show on Rajya Sabha Television. I'm Vishal Dahiya, and here we take a stock of India's fight against COVID-19 pandemic. Now today we're going to talk about Antisera. In fact, the premier organization, that is ICMR, in collaboration with the Hyderabad-based company known as Biological E.Limited, has developed a highly purified Antisera for COVID-19 treatment, although it is yet to be approved by DCGI and go for clinical trials. We'll try and understand for the next 25 minutes as to what exactly is an Antisera and how this particular development can help us further in this battle against COVID-19. And joining us for more on this is a distinguished panel of guests. Let me first introduce them to you, beginning with Dr. Samiran Panda from ICMR. He is Scientist G and head of the ECD division at ICMR. Also, we have with us Dr. Rakesh Mishra, Director of the Center for Cellular and Molecular Biology, that is CCMB, and Dr. Vikram Paradkar. He is a Senior Vice President and the Head of Technical Operations at Biological E.Limited, the company which, in collaboration with ICMR, has worked on Antisera. Let me begin with Dr. Panda here. Dr. Panda, let's first start by addressing the basic question specifically for our viewers. What exactly is an Antisera?
Well, good afternoon. This is a very interesting field and area of treatment. So far, human disease is concerned, and it has a long history, little more than 100 years. And the most interesting thing is that when the structure of antibody, these days antibody is a common household word. Everybody talks about antibody detection and all of these. But when even that was not known, what is an antibody or even antibody structure, the Antisera-based treatment came into being. So that makes it very interesting. 100 years ago, it is also interesting to recognize that from about 1901 to 1987, if one follows the development path of treatment based on Antisera or polyclonal antibodies or monoclonal antibodies, one could always track 5 Nobel Prizes given on this particular aspect. Okay. So understanding the disease dynamics and the treatment around it, when even antibody was not known to the scientists and the people in medicine, since then this has come into being. Now, what is antiserum? Serum is basically the fluid which gets expressed if we keep blood in a test tube. So there is clot formation, all of us know, because we have experienced cuts and bruises in our life, particularly since our childhood days when we used to play on the playground. But there will be a clot formation, and after the clot gets formed, there is a liquid, straw-colored liquid expressed from the blood which is kept in the test tube, and that's called serum. So in that serum, we have many proteins because the clot generally traps all the cells, be it red blood cells, white blood cells, platelets. So in that liquid part of the blood, what we get is antibody, which is produced by our immune system against infections that we experience. Okay. So that happens not only in human beings, but also in animals. So if the small animals or the large animals, they are injected with or infected with foreign elements, for example, a microorganism or antigen in smaller quantity, then it won't kill the animal, but it would stimulate the immune system of the animals concerned to produce antibody against it. So slowly you can increase the dose, and then if that animal which has been able to produce antibodies against a particular organism, if you challenge that animal with fatal dose of the organism, the animal won't die. So that was the observation. So historically, if an animal is able to protect itself against a challenged fatal dose of organisms, then could it work for human beings. And here we are talking about passively transferring that ability. So that's why the term which is being used for transferring antibody generated or developed in, let's say, equine, or which is horse, serum, and if we give it to the human beings, would human being benefit from it? So that's the whole concept. So it's a passive immunization, and it has worked not only for viral infections, but also bacterial diseases. For example, diphtheria or tetanus, these are the bacterial infections. And in viral infections, we have seen rabies, where immunoglobulins, generated against rabies is also used. So it is basically what vaccine does, but takes a little bit of time, but we cannot allow that window for the immunity to take over. Then the passive immunization actually manages the damage which could be done by the organisms, or the toxins which could even cause damage on a distant organ. So if I get an organism, it is not only at the site of infection that the disease manifestation starts taking place, but the toxin mediated injuries also hit the organs in the distant location. So there the passive immunization or Antisera generated in animals, or even more better if possible from humans, could prevent that sort of damage.
Okay. Let me bring in Dr. Vikram Paradkar here as well. Dr. Vikram, if you could take us through the work or the development which has been done on this front by your company in collaboration with ICMR, and where do we stand? What have we learned as far as COVID-19 and utilization of this technique in treating COVID-19?
Okay, thank you, Vishal. So this is a very good example of two organizations bringing highly complementary skills to develop a product. So when the COVID-19 infection started increasing, everybody realized that we require something which can give us a therapeutic option to bridge the gap between the time the vaccines will come onto picture. So this is a classic example of a therapy which is what we consider the antiviral therapy. So ICMRs and the NIV branch of ICMR, they already had developed the way to grow this virus. So they had isolated the virus, and they were growing this virus, and they also figured out a way to inactivate the virus. And once they did that, they approached us. We were also waiting for that collaboration opportunity. And they could give this inactivated virus to BioE, and we knew how to take that virus and develop an Antisera product from it. BioE has a long history, almost 60 years old history of developing Antisera products for various pathogens that Dr. Panda had already described. And so as soon as this virus was given to BioE, we immediately developed a way to formulate it with an adjuvant and then inject the equines. Mm-hmm. And so we already had the entire system set up. And as soon as we got hold of the right inactivated virus, we immediately started working on it. We already had experience of developing Antisera for various other products. So it allowed us to quickly get into equines. And by end of June, we already had first indication that the animals were responding. And when we sent our samples to NIV, they showed that this Antisera that we had developed definitely had the antibodies that could recognize the virus, that could bind to the virus, and most importantly, it can actually neutralize the virus. So as soon as we had that information, we immediately then developed a manufacturing process to process these antibodies. There is a enzymatic process that you need to follow to make sure the antibodies are highly purified, and they are formed in a formulation which is very purified as well as has a very consistent properties. And so in the month of July and August, we developed it, and it was tested at NIV to demonstrate that we did this process, which can give us a highly purified antibodies product, which can recognize the virus, and secondly, it can actually neutralize the virus. Based on that, we have now developed a product, which we are now ready to start clinical development to demonstrate that this product can actually give the benefit the patients,
Okay.
the COVID-19 patients.
Okay. And what's the status on those clinical trials, the phase 1 and all that? What's the timeline there we are looking at?
Yeah, so we have developed a clinical trial protocol. We have developed a product which is standardized, and we expect to start these clinical trials imminently this month. And once the trial starts, I think it will take us about a month to understand whether the product works in the target population. We expect to start it what is called the moderate COVID-19 patient. These are the patients we expect to get the highest benefit from this product, where this antibody dose will give the neutralizing power similar to what a convalescent plasma can potentially give. So this is our aim of our clinical trials. We expect to start them very soon.
Okay. Let me bring in now Dr. Rakesh Mishra. Dr. Mishra, you heard both the gentlemen here from both the organizations which have worked together on this particular aspect. So how do you view the development here, specifically the use of Antisera, the way Dr. Samiran Panda also described? I'm pretty sure you must be aware of those contexts out there. But in these pandemic times, this has been one more option to battle COVID-19.
Yeah, so first I would like to congratulate Biological E for developing this product. It's a very important development for fighting this big problem that we have, and gives confidence in people that there is one more arm available. Vaccine is quite far and there is no specific drug. So at least here this is something very specific. For the moment we are trying drugs which are antiviral, but they're not specific. So some of them help little bit, but most of the therapy is based on some steroid or some blood thinners, and those are secondary management kind of thing. But we have made a lot of progress. Medical care of the patient is pretty good compared to what it was 6 months ago. So that's why we are doing much better. Coming to the Antisera, it is really a good substitute of plasma therapy that people are using. In plasma therapy, we have issues like you have to pump in a lot of material, 200 ml or something, and so along with the possible antibodies that the patient has fought, from which you have taken the sera, also has probably other infectious agents or other chemicals which are not fit for the patient, who is already struggling very hard, because that's the time these things are given to the patient. So in such a situation, a relatively established and purified system like antibody treatment is absolutely far superior option at this moment. And here you see an example which is now ready to go for patient clinical trials. I'm sure that there are other efforts going on in direction. The only limitation here is the number of animals. You cannot produce them in factory, so just bleed the animals and collect them. So there is always production, but companies like Biological E and others have equipped themselves to produce in large quantity.
Okay.
The nice thing about this antibody thing is that, as you just heard, it is not the serum. You purify the antibody first, and then not only that, you can actually cut off the unnecessary part of the antibody and use only what is called fragment antigen binding, which is called FAV fragment. So it's very specific part of the antibody that you are giving to the patient. So this is like a bullet. It will go and straight away hit the enemy. In this case, it's virus. When snake bite, you give to immediately rescue the people. So anything that is foreign, against which you have made already the antibody, and if that foreign element is troubling body, you can inject and get the immediate relief. So I'm sure if this antibody, as you just heard, is neutralizing, doing well in the neutralization assays, which means is able to recognize and inactivate the virus. So that's something very good, and it will certainly be very helpful, because this problem of COVID is not going to go away easily. So we are going to face this for some time.
Okay, definitely. That's a very important element out there which you brought out, Dr. Mishra, and I'd like to bring in both Dr. Bikram and Dr. Samiran on this aspect. Dr. Bikram first, as Dr. Rakesh Mishra pointed out, this might be a good alternative as far as plasma therapy is concerned. Do you agree, Dr. Bikram?
Yes, so both of them work on the similar principle. You are looking at a source of antibodies that can recognize this virus and then neutralize the virus. Now, the problem is, where do you get the antibody from? So, what convalescent plasma, you are relying on patients who have recovered from the. The problem is, our human body responds differently, and probably not every person responds the same way. So, there is no guarantee that when you get a convalescent plasma, you are going to get enough antibody in that plasma, and you may not even have enough quantity of that plasma. While in this case, we develop a product, we test it, and every single lot we produce meets a specific requirement of neutralizing power. We have standardized that neutralizing power, which is going into a patient. And so that is the one advantage of Antisera therapy. The second thing that Dr. Mishra mentioned is the product we have made is highly purified, highly concentrated. So the quantity you have to give is not very large. You don't have to give hundreds of milliliters of plasma. And so I think the biggest advantage is standardization as well as consistency of the product. And the key is to develop that. And so that is where expertise that BioE brings into picture, expertise that NIV has in giving a standard virus formulation to us, that comes into picture. So that is how we have developed, and we hope in clinical trials we'll be able to demonstrate the consistency of the product, giving a consistent benefit to the patient.
Okay. Dr. Samiran, your views on that, because if I recollected rightly, ICMR's study on convalescent plasma therapy had, effectively concluded saying that it does not bring down the morbidity levels.
Absolutely. So as Dr. Bikram said, it's a bit unpredictable. It's basically the source of antibodies that one is looking out for. So one could be the animal source, like horse, equine serum used, or human. Now, if we take a little bit of time and try understand what happens when we take antibodies from a recovered pool of human beings who have experienced and have successfully cleared off an infection. So now one has to be then careful about not transmitting other infections that the donor might harbor. For example, in the past, hepatitis got transmitted when that kind of therapy was taken in because of the donor being a chronic carrier of hepatitis. So one has to look into that. But the advantage is it is a human source, so the adverse events or the reactions would be much less if another human recipient receives it. But the considerations from the animal sources would be side effects, because it's animal source, it's not human. But then, as Dr. Mishra rightly said, now these days we have mechanism to sort of cut out the frills, if you like it. So take just that bit which you need. And so that's possible, as Dr. Mishra very rightly said. And so you take that part, and that is the purified component which you would require. And then in animal model and other animals that you use, you can have a standardized approach towards not only development of the antibody, not only purification part of it, but also sufficient concentration, which is the issue. Because for convalescent plasma, we realized that it did not really prevent the progression of a moderate to severe stage of the disease. So that's why I would say that this collaboration and the product that has come out is one more that India could be, proud of globally, number one. And number two, I would say three things basically in this. What is the current commercial capacity for production of it? The second bit is the regulatory practice, the existing regulatory practices. And the third bit is the biologics and the science of the biologics. So this is fantastic development, and we must try see how does it fare in terms of its actual application. Of course, any Antisera could serve either as a preventive tool or a prophylactic tool, or and there is a history. let me tell you, take this opportunity to tell you that it goes way back during World War I, and NIH, that we are all aware of, National Institute of Hygiene in Health in US, that got established based on these kind of therapies, and there is a long history to it. And so this is the weapon, one of the armamentarium that has been added, as again highlighted by Dr. Mishra. So I think that given the current pandemic situation of COVID-19 and the challenges it has posed to human civilization, it should be considered as a great step forward.
Okay. One more question here, Dr. Panda, and that is about the future roadmap. As I asked Dr. Vikram there in terms of the plan for clinical trials. Obviously, the permission will have to be taken from DCGI, and then the process will begin. But since you spoke about the capacity to commercialize this and this being taken up as a good alternative to convalescent plasma therapy, provided the results in the randomized trials are very positive. So what's the plan there from ICMR's perspective?
That's a very good question. Actually, it's a very extraordinary time, if you may like it. It's an extraordinary time, and so it would naturally require extraordinary responses or the initiatives for innovations. But not that every innovation would hold true. But we have to try. And regulatory framework or the regulatory practices would also require very extraordinary steps or proactiveness. And we have seen that the DCGI, the Drug Controller General of India, and the regulatory framework in India is quite flexible and adaptive to the emerging needs of COVID-19 pandemic. So the way we see the whole story further unfolding over the next 1 month or so is rapid regulatory approval for taking it through a trial in real world setting, and then quickly try understand how does it help one individual who is infected with SARS-CoV-2 and should be hopefully out of the whole situation within 14 to 21 days. So the window is very small, and so that's why this tool, this particularly developed tool, Antisera, where you are actually passively trying to immunize that person to struggle through that critical phase, so that he or she does not move from the moderate stage of the disease to the severe stage, or rather go backwards from moderate, goes into a milder stage, and then finally, clears off the infection and becomes all right. That's the whole attempt, and I'm sure that the DCGI and the existing regulatory framework in India would allow a fast, undertaking of the clinical trials. Of course, we have CTRI, Clinical Trial Registry of India, and any trial that happens in the country needs to be registered on that. And ICMR will be more than happy. ICMR not only maintains this platform where anybody can register, but ICMR will be more than happy to join hands, because that has been our track record. And we do support any kind of initiative, be it diagnostics, be it treatment, be it prevention, technologies which would help the country and ultimately the countrymen, all in India and outside.
Okay, definitely extraordinary times does require extraordinary efforts. One last concluding comments from Dr. Rakesh Mishra here. Dr. Mishra, how do you view the situation developing here? We have a renowned, public-funded organization lining up, teaming up with a renowned private sector company. Obviously, it looks like the results are terrific here.
Yeah, this, like Vikram said, this has been an extraordinary collaboration where distinct organizations come join hand with multiple, with complementary expertise and deliver something which is useful for the society. So that's certainly, and the country has really responded well. And this is one of the reasons how we have managed so far pretty okay this disease, considering the dimension and considering the damage that is caused to other countries. I mean, there are economic consequences which scientists or clinicals cannot control. That is something else. But disease management has been quite fantastic from this point of view. I just wanted to just add that in these kinds of times, when you make these kind of molecules, these kind of biologicals, and you test them, and one more thing you do is what is called safety experiments, in which you show that if you inject to small animals, it doesn't cause any damage to them. And then you can, if there is possible, to infect those small animals and show that it does not affect them. So these are very fast. These are called pre-clinical trials and safety trials. These can be done in the labs in a couple of weeks or 3 weeks. And with all that, if you go to the CGI, in considering the emergency situation, if it's required, they may give you special permissions to use. So these are things which give us a big advantage of helping out people who need a little bit of tilt to survive. And these are something they will be very useful at that time. And it's very likely they will not be harmful because it's something very specific which is made against a particular virus. So it's going to be a specific, generally speaking. So I'm reasonably sure that this will see through the basic requirements, and considering the situation, it will be useful for the people. And I see no reason why DCGI will not facilitate it in a fast track and emergency provisions.
Okay, there it is. Thank you so much, Dr. Rakesh Mishra, Dr. Vikram, as well as Dr. Samiran Panda, for sharing your views with our viewers. This is all about Antisera, being developed by ICMR in collaboration with Biological E.Limited, a renowned private sector company. The idea here is to go ahead and find a cure, a treatment for COVID-19, a solution for the problem which has been thrown at us by this particular pandemic. We will have to wait for randomized clinical trials to happen and their results to come in before this particular therapy can be utilized. We'll come back again and we'll bring more details on this aspect as well. But till then, you keep watching Rajya Sabha television, and please do not forget to wear a face mask when you step out of your houses. Thank you.